current studies are really focused on the mechanism for amyloid protection. Until recently, the prior publications, we looked at the forms of metals. We knew in vitro that amyloid-β suppressed copper oxidation and played a role in iron oxidation also. So we looked at the forms of iron and copper present in plaques. We were able to identify magnetite crystals within plaques, iron 2, iron 3 oxides by using high atomic resolution microscopy. And in a more recent study performed with Joanne Collingwood in England, we were able to actually define the oxidation state of both iron and copper within plaques, and found very surprisingly, we were mainly looking for, can we find copper 2 and copper 1 redox clusters, which would show that there was active, the copper was active in redox chemistry in the plaque. Well, we found that. But the more surprising finding is we found that there was a dominance of metallic copper and a dominance of metallic iron. Neither have ever been found in the human body before our study. In the case of iron, to our knowledge, this is the first description of iron ever in a biological system. Metallic iron. Why? Metallic iron is super unstable. The rusty nail. But remember, this is atomic iron, so it's much more reactive. And also our study was quantitative, and so the dominance of these metals actually were in the metallic form. Why have they been missed before? Why didn't anyone see them? Number one, they didn't use the same techniques we looked at, because the techniques we used prior to this were all based on looking at redox active forms. Metallic metals are not redox active. They actually are out of that sphere. And so it could be that they're more common than we ever thought, because nobody put an X-ray microscope hooked to a synchrotron before, or it's really unique. And we're expanding that observation. So there's two places we're now expanding it to neurofibrillary tangles, and that is ongoing. And the other piece that we're doing is trying to understand the mechanism for production. In the case of copper, it has been known in biological systems before, but in bacteria. So there are mechanisms that could be important in generating it. So that, and why are we doing this? We're trying to understand how amyloid really works. We want to understand how the plaque really works to reduce oxidative stress, because we think that it will provide a new avenue for therapeutics. And to work with the biology, not against it.